Namaskar दोस्तों, welcome to channel Toxicology on board. Today in this video we'll be discussing another DABT topic that is reproductive assessment by continuous breeding RACB protocol. I will be discussing with respect to NTP. I will also request you to list out all the OECD guidelines with respect to reproductive toxicity, like first generation toxicity or second generation toxicity, and prenatal postnatal toxicity PPNT studies, which is the OECD guideline responsible for this type of and combined reproductive and repeat dose toxicology study. What is the OECD guideline number? So all the guidelines which respect to reproductive toxicity, right from segment 1, segment 2, and segment 3, that is fertility and early embryonic development, then embryo fetal developmental study, which include teratogenicity and prenatal postnatal developmental toxicology studies. In extension to that, you have also read about juvenile toxicology studies, the one which the guidelines which are OECD guidelines which are responsible for this type of this thing. And with respect to ICH also, read ICH S5 guideline, that is reproductive developmental toxicology studies. ICH not ICH S6. ICH S5 is for biotechnology derived guidelines. For ICH S6 is for your reproductive developmental toxicity. So they have listed some drugs under ICH 6 guideline, which are having MEFL value, that is maternal embryo fetal developmental toxicity limits will be there. So read those things, which are very important with respect to hazard identification for the risk assessment toxicologist. So in this video, we'll only discuss about RACB protocol. Uh, Other type of things, there are some previous discussions have already been done in the channel. I'll try to give those things in the video links in the description, and other things, list of OECD and ICH guidelines which I told you. You yourself make some notes so that you can compare and read these guidelines. Okay, so we'll discuss RACB guideline, which is breeding protocol, which is given by NTP. NTP is National Toxicology Program. So first we'll see what is this RACB. So whenever you do some risk assessment on a certain chemical, when you do the literature search, you'll come across one type of studies called RACB protocol studies under reproductive studies. So that we are discussing here. Reproductive assessment by continuous breeding, RACB in short form, is a standardized toxicological protocol developed by the National Toxicology Program to evaluate effects of chemicals on reproductive system in the case of rodents. Here the test species will be mice or rat. Okay, unlike conventional multi-generational studies. So as I told you, segment 1, segment 2, segment 3, you will also have multi-generation toxicology studies also in order to identify the reproductive hazards. RACB utilizes a continuous cohabitation period to assess reproductive performance over time under a constant challenge of the test substance. Okay, so here continuous cohabitation is the main key thing that you have to remember. What is the core protocol of the structure? We'll see F0 generation continuous breeding. Okay, this is first generation, sorry, zero generation, not F1. A pair of animals, typically Swiss CD1 mice, is housed together and exposed to the tested substance for a 14-week continuous breeding period. Okay, then next, multiple litters. During these 14 weeks, the pair typically produces 4 to 5 litters. So the group of mice which are born, no, it is called litter size. So this is one of the things in the case of reproductive studies you will be measuring, one of the parameter you can say. Crossover mating. If a reproductive effect is observed, a crossover mating trial is conducted using treated and controlled animals to determine if the male, female, or both are affected. Okay, here in this case of crossover mating, you are doing crossover mating with using treated and controlled animals. Next, coming to F1 generation assessment, the final litter, usually the fifth, is often rare to maturity to evaluate the reproductive performance of the second generation. Key assessment endpoints. The RACB protocol monitors several sensitive indicators of reproductive health, fertility and fecundity, number of litters per pair, number of live pups per litter, proportion of the pups which are born alive. So these are the various parameters that you will be measuring in the case of this RACB continuous breeding protocol study. Time to litter measures the latency of the parturition, which can indicate subfertility or delay in the case of delivery of this particular pups. Pup data. Live pup weight, you will measure the weight of the sex ratio how many males are formed, how many females are given birth in that particular litter, survival through postnatal development, even after the delivery of the baby, how many days it is taking to survive, or there are any certain pups which are which undergo mortality on day 2 or day 3. So till the postnatal development will be checking the survival rate of the pups which are delivered. Next, adult necropsy include organ weights, testis, epididymis, ovary, sperm parameters like concentration, motility of the sperm, morphology of the sperm, is the head there or not, and estrous cycle evaluation. So these are all the assessment key endpoints which are measured during this RACB protocol study. So what are the advantages when compared to multi-generation study, when compared to segment 1 or segment 2 or segment 3? Why RACB protocol study is having more advantages when compared to multi-generation or other type of reproductive studies we'll see. Increase the sensitivity. Why? By using this RACB protocol, by producing multiple litters, up to 5 generations we are seeing, the protocol can detect subtle changes in the fertility that might be missed in a single mating study. In the case of, if you look into FEED study, that is fertility early embryonic development, single mating means only one generation is done. So anything which is missed during this type of single individual mating study, that can be captured in the case of RACB protocol, because of which this is having more sensitivity when compared to multi-generation or segment 1 study. Temporal tracking. This RACB study, it distinguishes between early effects and progressive toxicity. Okay, so is there over a period of generation, is there any toxicity is there that is that develops over long-term exposure, or is there any uh, early effects of progressive toxicity that can be detected by using this RACB protocol. Next, efficiency. The RACB design generally requires less time when compared to multi-generation study and fewer animals than traditional EPA and FDA multi-generation protocols, while maintaining the high statistical power. Since we have huge number, up to 5 generation is there, n size is more, population size, litter size is more, n size is more. So because of the high statistical power, the data generated from this RACB protocol will have greater advantage when compared to FDA or EPA multi-generation toxicology studies. So what is important here is you have to remember, for see, being a toxicologist since 8 to 9 years in the industry, I do not come across too much of studies, to a great number of examples with either drugs or chemicals where they have RACB protocol study or data. If there is a certain chemical which is susceptible to cause multi-generation toxicity, then only a particular toxicologist or scientist will do a RACB protocol study. Another thing is that NTP. If NTP is after doing, when it identifies a certain chemical, that this particular chemical is having hazard and it is having some hazardous, if it is this particular chemical is exposed to human population, if it is a possibility of causing hazardous adverse effect, then at that time this NTP will do this type of RACB protocol studies in multi-generation. So if you look into NTP toxicology program, that NTP report, if you see a toxicological report, you will see you will have a dose ranging finding study will be there, that is 14-day study, 13-week study you will be having, and 2-year study you will be having. In that you will be identifying whether that particular chemical is causing any cancer or non-carcinogenic, that you will be seeing. Okay, so NTP reports also you see, and pause here, also try to go through this particular diagram. F0 generation, F1 generation, F2 generation, at what point cohabitation is there, at what time dosing is there, at what time, what are the different parameters they are measuring, like anogenital distance, litter size, pub size, uh, these parameters what they are measuring, that you can go through. And another picture I have taken it from handbook of toxicology. This particular thing, in the second edition, page number 494, they have given a schematic diagram of RACB protocol. This gray areas are cohabiting studies, and these are the dosing days. So pause here and read about this particular cycle. Okay, so hope from this video you were able to learn about RACB protocol. See, reproductive toxicity is very, very important in the case of risk assessment, hazard identification, whether a certain chemical is having hazard, reproductive hazard, or teratogenic hazard, or maternal toxicity, fetal toxicity, or embryotoxicity. These kind of flags you need to raise. So for that, you need to have either segment 1, segment 2, or segment 3 study, or prenatal, postnatal, or juvenile toxicology study. So in that, and you have a separate guideline for ICH S6 guideline is also there. So in the this is very important. Not many of us toxicologists will have an opportunity to work in reproductive toxicology study as a study director or study personnel. So understanding these type of reports is also very important, because being a risk assessment toxicologist, without doing the actual study, you need to identify. For that identification, first you need to understand the study plan, study report. For that, you have to understand the guideline based on which this particular study was conducted. So thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. All the best for your exam.